Greeting, greetings, and welcome to What's Up in the Watershed. I'm Kalita Scott, and I'll be your host for today's question and answer podcast. Today, we're being joined by Nicholas Carteau. He's a research technician interested in expanding knowledge in marine ecology. He received a Bachelor of Science from University of South Florida and is currently pursuing his Master of Science degree from Unity College. Nick has spent time in conservation, research, and operations with a focus on ocean and sea turtle conservation. His current research interests involve focusing on oysters, seagrasses, and mangroves, and understanding the role they play ecologically in our own backyard. So excited to have Nick here. Welcome.
Thank you for having me, Kalita. I'm super excited to be here today. And that was a very wonderful introduction that I'll just jump right
I've
into.
got a little bit more. I want to give
Oh,
everyone some time to get on.
oh yeah.
Um, But I just want to say that one of the great things about science, I feel, is that experiments spark new questions, and that curiosity drives progress. So today, we're going to get this great update from our partners at Florida Oceanographic, and they're playing a key role in the IRL NEP's lagoon-wide restoration network that focuses on seagrass recovery. So we hope that everyone can learn from their experiences, the successes and the challenges, as we all continue to work together to support the Indian River Lagoon. Take it away, Nick.
Thank you for that. So that was a very eloquent introduction. So I will just jump right into it. I am a research technician, but I'm also the nursery manager here at the Florida Oceanographic Society. And today I'm going to walk you through our seagrass nurseries, their importance in research, conservation, and collaboration. So a brief overview before we dive into it. So I'll be kind of going over seagrass as a whole to begin with, for those who aren't necessarily familiar with it. Our seagrass spaces at the Florida Oceanographic Society, with a special regard to the past, how we got started, the present of what's going on now, and then our future plans as well. So seagrasses, to begin with, are underwater plants specifically adapted to grow in salt water. That's a unique attribute that not many other plant species are able to replicate. They are also considered a foundation species, so they serve multiple purposes. They form habitat for other species, especially those in their juvenile life stages. They are a primary food source for a variety of different organisms, and they influence the chemistry of the environment around them in the form of oxygenating the water and changing the sediment around them. There are several different species of seagrass spread throughout the state of Florida. However, the main three that we'll be focusing on today for Florida Oceanographic sake will be Seagrass filiforme, which is manatee grass located in the top right corner here. Hopefully you can see my mouse. If not, I'm just going to talk out loud about it as well. Halidule wrighti, which is shoal grass located in the middle picture here, and Thalassia testonium, which is turtle grass located in the bottom right here. So you can see with each of these three species of seagrass, they are very physiologically different from one another. All seagrasses, however, suffer from anthropogenic influences in the form of freshwater discharges, algae blooms, and poor water quality. An example of this is a comparison of the years 2016 and 2017. So you can see here in 2016, around these barrier islands here, where the St. Lucie estuary meets the southern part of the Indian River Lagoon, you can see all these dark patches are seagrass meadows. However, when we fast forward to 2017, you can see that the vast majority of these patches are gone, and it's mostly just bare sand. So seagrass species aren't the only organisms affected by poor water quality. It can also affect oysters, gamefish, and other species that live or are adjacent to these environments as well. So the overall role of nurseries is to establish stable stock of various seagrass species for research and restoration purposes. So with that in mind, I will actually dive into Florida Oceanographic. So I'm going to actually show you what our facility looks like. For those who have not been here, I highly recommend that you do please come. We have a variety of different habitats and frames that you're able to learn a whole ton from. And one of these spaces that I'll be focusing on in great detail will be our Gamefish Lagoon Aquarium. So this space is this big pond area that you see here. It is a 750,000 gallon aquarium that's fed by fresh salt water from the Atlantic Ocean from an inflow pipe. So the water enters this space and flows in a counterclockwise direction, And this is important because this is where we get our water for our seagrass spaces. And we're going to talk about our seagrass spaces now that we have, starting with our seagrass meadow, or what I call the meadow. So this is basically a retention pond located on the western part of the Gamefish Lagoon. So how the water flows into this space is it enters along this part here, and then actually just flows out through the drain pipes located on the north and south side of the meadow. So this meadow, you can see all the dark patches there, that is all seagrass. So this is seagrass that I'll be focusing on in more detail with its origin, but we use it for our research projects that we have, as well as different restoration projects as well, like our seagrass matting events. The second space that I'll be focusing on too is our front of house space. So this is 34 110-gallon flow-through tanks connected to our Gamefish Lagoon on a circulating system. So the water flows through this system a little differently than our meadow. So you can kind of see there's a white pipe there, that's our inflow. And so the water from the Gamefish Lagoon enters through there, goes through all of these tanks that you see here, and then exits back into the Gamefish Lagoon. The third and final space that we'll be focusing on towards the end of this presentation will be our back of house or experimental array. So these are 4 tanks and 4 blocks that will actually function as our experiment space. And I say will because we are currently under renovations with that right now that I'll be focusing on towards the end. So that's how all these spaces are connected and what their purposes are. However, what you're probably wondering is, where did we get all this seagrass from in the first place? So we have our FOSTER program to thank for that. So FOSTER stands for Florida Oceanographic Seagrass Training, Education, and Restoration. I know, it's a mouthful. So I usually just refer to this program as FOSTER for short. So how it works is, over the course of several years, staff and volunteers from Florida Oceanographic actually walk along the shoreline and look for seagrass fragments. And seagrass fragments look like the one picture that you see here to the right. So it's a shoot and root structure that is still fully intact. You cannot have one versus the other, otherwise those fragments are not viable. Once these fragments are collected by the staff and volunteers, they are placed in wet buckets and then brought to us at Florida Oceanographic. These fragments were replanted and are still growing within our nursery tanks in our meadow. So all the grass that you saw from those images and all the grass that you see in these tanks here all started out as fragments at one point or another and have just grown out over the course of the years. These individuals can be viewed by the public, so these spaces you can actually walk by and actually see firsthand. And they serve as wonderful education tools for anyone visiting our facility. So I do want to give a shout out to our wonderful volunteers as well, for it, if there are any on here. If anybody understands that you cannot do this without people to help you, they are wonderful. So for example, the volunteer that you see in the middle picture there, her name is Star. She's been helping clean our seagrass tanks in the front of house for years, and without volunteers like her, we would not be able to maintain our nursery spaces. There we go. So with that in mind, you can see on the map here that we have a variety of different locations that we have potentially collected from. So that in turn means that we have a potentially high genetic diversity from the fragments. However, one of the main questions we have is, where are they from? So as we know, currents ebb and flow and tides rise and fall. So these fragments may have not come from areas that we think they come from, or maybe from even further away because of that. So to answer this question in regards to manatee grass, we are actually working in collaboration with Dr. Seagrass Garcia and Dr. McCoy at Harbor Branch Oceanographic Institute in a project funded by MOTE, which collected several samples of manatee grass from several wild areas that you see represented by the orange dots here. But along with those several wild areas, we actually collected from 2 seagrass nurseries, so Florida Oceanographics and Harbor Branches. For those who don't know, Harbor Branch also has a seagrass nursery as well. So yes, very scary image here, I know. I promise I'll walk everybody through it. So this is a dendrogram, for those who aren't familiar, and the horizontal lines indicate the distance between connected groups, and groups are made of individuals collected from different populations. So to simplify it, the groups located in this circle here are more closely related, And these groups in here are more closely related to each other as well. So that's why there's two distinct circles kind of separating the two. Focusing specifically on the nurseries now in this part of the dendrogram, you can see that Harbor Branch is represented by the blue circles here. So the blue circles are actually closely related to samples collected from Jim's Island. So that's a region kind of close to the Harbor Branch nursery. However, with Florida Oceanographic represented by the black triangles, you can see that in this one here, they're more closely related to samples collected from Sebastian and JC, while these ones on this part of the dendrogram are more closely related to samples collected from Waxahachie. So if you know your geography, you know that those 2 points are very different from one another. This population here, we don't know where it's actually what it's related to, because out of all the samples we've collected, it did not match with it, or was related to any. While this one up here was more closely related to Lake Worth. So what does this all mean? Breaking it all down. Florida Oceanographic has genotypes from several different regions in the IRL. So from the kind of northern part of the IRL to some really southern parts of the IRL. They might be locally adapted to different stress testers. And what that means is, for example, samples from Waxahachie, for instance, just to hypothesize, might be more suitable to higher temperatures, or samples from Sebastian and JC might be more tolerant to salinity changes. And this is exciting in the fact that this opens up the door to more future experiments, now knowing where these samples are and to see if they are locally adapted to certain stressors. So this also helps us with informed restoration. So it can either increase the diversity of the rest of our plots when we do restoration. So for example, now that we know that certain ones are from certain areas, we could combine those ones to see if that actually helps with the transplantation success. Or we can maintain the locally adapted genotypes. So now that we know certain ones are from Waxahachie, we can continue to put those ones from Waxahachie back in that region. And the same thing with Sebastian and JC. So this is actually what it looks like, the genetic samples. So we actually have them in one of our tanks in the front of house, and they all have these green tags that you can see in the picture here. So this helps us identify where each one is from and without losing track of them. So this sequencing of the genome from manatee grass will allow us to understand what genes are different between populations. And in addition to this, we will also be doing the genome for Halidula or shoal grass, which we will have rad sequence data that compares the grass that has survived a mass mortality event back to the source population in our nursery. So this is helping us create this much fuller picture of the origin of the grass, where it's from, and how that's going to help with our projects, experiments, and restoration going forward. So I've been focusing a lot on the positives of how all this got started. However, there were some drawbacks of our old design. Starting with the middle picture here, as you can see, the seagrass is in this plastic tray. So if anybody knows anything about plastic, plastic does not last long in environments that have salt or sun. And what was occurring with these plastic trays is chunks of them would actually break off and float to the surface. And if they were not collected from our tanks, could potentially end up back into our green fish lagoon. And that is not a good thing, because the organisms in that environment do not need to be ingesting plastic in any way, shape, or form. Another drawback is it reduced the growth of the grass. So when the fragments were initially collected and planted into these trays, they would start to grow out gradually and reach a point to where they would hit the walls of the tray, and their root structures would actually grow out of the tray and become exposed. So this would actually stress out the plant as it was trying to grow out of it, and we would actually see grass die back and die off because of it. Another important thing is we do not have any uniformity with our tanks. So you can see in the image on the left here, there's not an optical illusion. Some tanks were actually taller and some tanks were actually shorter. And what we experienced with the shorter tanks is because of how low the water height was and how tall some of the grass gets, the tips of the grass would actually become sunburnt due to too much sun exposure. So now fast forwarding to the present, we still have a total of 34 seagrass tanks, and for those who are interested in the specs, I always like to include them just because I'm a detail-oriented person as well. And they all have a PVC standpipe drain at one end and a seawater inflow at the other side. But however, each of these tanks aren't all serving the same purpose. 22 of them are functioning as grow-out tanks, which are designed to focus on the growing out of specific species, while 12 of them function as experiment tanks. With the grow-out tanks, we had a specific focus on these three components: sediment height, composition, and water flow. So sediment height, we kind of went back to the drawing board. So once we removed all the trays and started to actually add the sediment, you can see here that it's at least 4 to 6 inches. So that's drastically a major improvement compared to what we had in the trays, which is usually about 2 to 3 inches. The composition is something else we also changed. So we actually got rid of all the old sand that we did have in those trays. And going forward, like in the tank that you see there, we actually did a mixture of live sand, clay sand, and aragonite. So we definitely diversified what the sediment composition was. And water flow was a little bit more of a tricky situation to figure out that I'll definitely be getting into. So seagrasses in their natural environment experience a lot of wave motion and just general energy that they have to be exposed to. So this seagrass is turtle grass, and as you can see, it is very much exposed and very much moving around with the wave action, which is that's how healthy seagrass is supposed to grow. So when it came to kind of mimicking what's out there in the wild, we had a little bit of trouble because our inflows, as you can see here, just drop into the water and then flow on the surface and flow out the drain at the end. However, to solve this, or at least to solve this initially, I tried to come up with what's called a T-bar. So you can see attached to the inflow here, I put something that's like a reverse T under the water, and under the water you can see there's several different holes pointing in one direction. So when the water turns on with the inflow, it would actually flow under the surface of the water and actually move it under the surface as well. However, one of the main drawbacks of this T design is it just didn't move the water enough. So it moved it a little, but just not simply what we were desiring. So had to go back to the drawing board once again. So for those who want tanks that have who need a lot of wave motion, I would definitely recommend this. So this is what we call the Octo Plus. So this is a wave maker. There are two attached to this circuit board here, or this controller, and they actually have several different settings. So they attach themselves onto the sides of the tanks with a magnet that you can see here, and you're able to adjust them on the height, the speed, anything you could possibly imagine. So this is what it looks like now. So what you see here is all the water motion is being generated by just those two wave makers. There's no water entering from the inflow whatsoever, and that's much more of a significant flow than what we were experiencing. And because of that, we've actually seen the grass increase its growth due to the kinetic energy that it's getting from this wave motion. So we altered the sediment height, composition, and water flow. So those are the big three that we definitely we nailed finally. We are also interested in species interactions though. So along with considering those abiotic factors, we're also considering biotic factors. And that's what we'll be getting into with our experiment tanks. So this is actually an active experiment we have running in those 12 tanks I talked about before. So it includes fighting conch, wave makers, and a combo of the two. And the purpose is to see how those factors, the abiotic and the biotic, are affecting the growth with natural stressors. So we're kind of mimicking what the grass would be experiencing in the wild when it comes to species interactions and motion from waves. So I know it's a lot. What has changed? So we've refurbished all the tanks and the tables. So along with replacing the wood, it's now made of composite material, and the tanks are all the same height and measurements now. They all got replanted into sediment, not plastic boxes. So that's a big plus in my opinion. And all the sediment got replaced. So that's that mixture of clay sand, live sand, and aragonite that I talked about before. We also added electricity to the space. So that's what's able to allow us to have those wave makers that you just saw before. And we're also adding biotic and abiotic realism to these tanks as well. So with these renovations, this is kind of like a personal goal slash department goal, I would say. It is to double the stock of all the current species we have. We also want to add new species of seagrass that we don't have currently. And we also wish to increase the research collaborations with other nurseries in the region, not just Harbor Branch, but other ones as well. And also to increase nursery sales, because I always like to say this, we do sell seagrass. I know that's a very niche market, but for anyone who's interested, we're definitely open to anything. And then lastly, we're also creating the genetic libraries of our local species as well. So with all that in mind, that's a lot to stay on top of, I know. So you can see here in the bottom left image, this is what the front of house nursery currently looks like now. It's very much uniform and clean and just, it's a well-oiled machine. But to keep that well-oiled machine running, we have to keep things in order. So to do that, we have this wonderful checklist that we have in our lab here. So that's color coded to represent certain tasks that occur during certain times. So for example, the red marks all the daily tasks that need to occur. The green marks the weekly, and then the blue marks the monthly. So I will not go over every single task, but for example, a monthly task would be cleaning out the drain pipes that lead up to the Gamefish Lagoon, while a daily task would be just checking the wave makers and making sure that they're all running properly. So this is my kind of segue into we sell seagrass. For anyone who's interested in seagrass, contact is initially made through email. So if you're interested in having it for an experiment or some other purpose, you reach out to us and let us know how much you would like if you need a full bucket or anything less than that. And then once we figure out the details, it is followed with an in-person pickup. So you come here and we actually provide the seagrass to you in a secured container with salt water. So the reason for why I say that is we do not transport any seagrass without any salt water due to the fact that the grass would become dehydrated and we'll see increased mortality because of that. So another point I'd like to focus on too is our collaboration. So I've talked a great deal about FAU and Harbor Branch already, but another thing that we've also been collaborating with them with is we actually utilized an experiment in our meadow space that focused on the conchs and along with uh, sediment samples out there as well. Another group that we're also working with is University of Florida with an up-and-coming project next year that will be doing a co-restoration with clams and seagrass in our part of the IRL. And then if anyone has any more questions about this last part, Kristen McCoy will be more than happy to answer any of them. I'm not too familiar with all the details of this yet since we're just getting taken off with this. We'll be doing a collaboration with FIT as they are going to be using some of our seagrass for a growth chamber study, specifically focusing on biochar, which is carbon. So the future. So for those who've been waiting for this, this is what we call our back of house space or experimental, right? Like I was saying before. And this helps break down what the setup's going to be. So you can see there are 4 tanks and 4 blocks, so a total of 16 tanks. And each one of these is separated on its own circulatory system, so its own inflow and outflow, so they are not connected to any of the other blocks. And this is significant in that if we want to do any research, or if any other researchers that are visiting us wish to do their own experiments, they are able to do so. So for example, if one researcher wants to use 2 blocks and another researcher, wishes to use another 2 blocks, they are more than able to, so without interfering in any of their experiments. And with that in mind, I am at the end of my presentation. I figured I would end with a wonderful picture of our meadow. So this is a up close and personal view. So all that entire dark patch that you see there, that's all my wonderful seagrass that I spend so much time and effort focusing on growing. But if anybody has any questions or comments or concerns, I'm more than happy to answer any of them now. Thank you very much for having me.
Thank you for joining us, Nick. That was comprehensive. Thank you. Thank you. We do have a few questions.
Sure.
The first one in the chat, or we got a couple. Can you explain why kinetic energy helps promote seagrass growth?
So just like most terrestrial plants that you have in your garden or that you see around in a forest, kinetic energy kind of is a stimulant. So like winds, like you wouldn't think a wind would actually help like certain trees grow, but it actually does. So it's the same thing with seagrasses, just under the water more so. So the wave action that they experience actually is a stimulant that helps them like helps their blades get more sunlight or helps the because you don't want them to just be in a stagnant pool of water the entire time.
All
So
right.
to answer that question, it helps. It's a stimulant that encourages growth, just like how herbivory helps them grow. When a manatee like takes out a chunk of the seagrass or a sea turtle, then that seagrass will actually grow back in fuller because it wasn't removed, and it actually encourages the grass to kind of go into overproduction of like photosynthesis and those other like biological factors as well.
All right. Thank you for that answer. And I've got a follow-up question to that. Does kinetic energy have an effect on pollen and seeds?
Yeah.
So yeah.
I could definitely say yes to that. A perfect example I like to use all the time is mangroves. So mangroves are a very unique plant in that their their seeds are what we call propagules, and each of the three species of mangroves that we have in Florida, their propagules look drastically different from one another. The one that most people have probably seen the most of is the red mangrove. It's that long pencil-shaped one. So how it works is kinetic energy, like wind, actually knocks those propagules out from the trees to begin with, and then oftentimes those propagules will actually land in the water, which is then moved by tides and wave motion, which actually brings those propagules which float on the surface of the water to entirely different areas. So because of that, kinetic energy is actually a major influence as where mangroves establish. So that's just one of the many examples in the plant world of how kinetic energy has a role in distribution.
Energy, always moving, so important. Another question, do you grow the same species in the meadow as well as in the tanks?
Oh, that's a great question. I love questions like that. So the meadow space is primarily shoal grass. However, we've actually been recently adding the other two species in there. So like we actually have a patch of turtle grass that's in there. So if you ever do come by and visit us, I always love showing people, oh, there's my turtle grass that I put out there. And we're doing the same thing with our manatee grass as well. So we're trying to make more of a mixed meadow situation out in the meadow space, just simply because that's how the seagrass grows in the wild. So the seagrass doesn't just grow like it's all one clump and it's all another clump. It's a healthy meadow is multiple species growing in conjunction with one another.
I co-sign that. Another question here, where in the Loxahatchee did your manatee grass come from? Is it the locations where the Water Management District and Wild Pine Lab water sampling occurs?
It
That's a
might be.
specific
I
question.
wasn't the one who collected those samples, so I'm not 100% sure. But I do know there's seagrass there, so if I had to take a gander, I would say yes to that question. But like I said, I'm not the one who collected those samples, so I'm not 100% sure.
Well, maybe we can follow up and get the answer to that.
Yes.
Um, Here's some more questions. How difficult is it to pivot once your project is underway? And then how do you weigh the risk of staying the course versus trying something new when something doesn't work?
So that's just the beauty of science, that nothing ever goes according to plan. I mean, a perfect example of that is we are actually do one of our interns is actually doing a bald cypress experiment where she's actually seeing how mangrove inoculant, which is essentially chopped up propagules, can actually help increase the salt tolerance of certain species. So she's specifically focusing on bald cypresses. So initially, that experiment kind of didn't get started right away because the plants that we did get actually all died before we could even get started. So then we kind of had a delayed start. And now, one of the more recent unexpected twists of the experiment is we've had animals actually eating some of the bald cypress leaves, which we were not accounting for. So that's just one of the things where we still have the plants are still alive, so we still keep the experiment running because we're still getting valuable data because of that. So that's just one of the many examples of a things not going according to plan with an experiment. And I always say just you always try to keep the course the best you can until it just becomes an absolute disaster and you can't save anything from it.
That's awesome. Thank you. A follow-up question from one of our guests. They're going to follow up with you about the sales of your seagrass.
So
They wanna-
fun.
Yes, they want to know about the BMP for outplanting and about the testing of seagrass and testing of seagrass and biochar media. So that might be something more specific to follow up with you and Dr. McCoy there later on. Do you have any insight on that?
We were just getting started with that. I was just kind of given the tidbits of it just the other day, so nothing, still in its infancy. So I would definitely, we can definitely circle back on that one.
Awesome, we'll do that. Let's see, one more question in the chat. Let's see. How do you keep people motivated when the plan has to change in a changing environment such as your center? How do you keep everyone motivated?
That that's a very good question. I like to think that I'm surrounded by a very passionate group of individuals in my organization. So it's just one of the things where you kind of, when you jump into this field, you kind of know you're selling your soul to the mission a little. That you're passionate about what you do and you deeply care. And one of the things that that Florida Oceanographic is kind of focusing on too, I would say now, is trying to do more of a like responsible restoration because we're realizing that unfortunately with a lot of our seagrass restoration projects, they don't always succeed. We have a lot of we had a perfect example of this where I was very much involved with a restoration project that we had on the back of our property in the past year or so, and then at a site at Fort Pierce. And unfortunately, we saw two different things occur at those sites, but the seagrass actually died off at both sites for different reasons. So one of the things is just kind of being aware of, okay, these things occur in certain environments. Like for example, with our Florida Oceanographic site, we had discharges. So that's something to definitely take into consideration when it comes to doing seagrass restorations. When they're going to do discharges, should we try to get the seagrass out there months in advance before that, or should we just not even try to do some of those sites near where they do the discharge events? Should we try going further north? It's just one of those things where that's kind of what we're doing now, and that's why we're doing all these experiments where we're doing the genetics, where we're seeing these species relationships with the seagrass because we're trying to figure out better ways to help this grass actually establish, and so we can increase the success rate of these projects as well.
All right, just had one more pop up.
So fun. I love questions, so this is my bread and butter.
Well, it's great. That's why it's so wonderful to have the people who are doing the science come share their insight. What new technologies or tools um, do you use or do you hope are to come available to help you overcome some of the challenges?
So we've actually been testing some of these new technologies. For those who wish to do their own research, we actually have been testing what's called these bees, which are these kind of like honeycombed, hexagonal, pentagonal shaped like structures where they're made out of potato starch, so they're like biodegradable. And we've actually been testing them out to see how the grass actually grows in the bees. Unfortunately, we're kind of coming to the conclusion that the grass really does not like the solid structure. So that's just one of the things that we're trying besides the normal like matting that we do. Because normally when we do seagrass restoration or when we put seagrass on in wild locations, we typically have it attached to these burlap mats with floor water. And however, the thing with the burlap mats is they take a very long time to actually biodegrade. So for the previous project that I was talking about, the Florida Oceanographic and the Fort Pierce sites, we
Mhm.
actually didn't see the burlap completely like disappear until like months after like we actually put the grass out there. And so that we think that might have been one of the components as to why the grass didn't immediately establish itself because it was still trying to fight through the burlap a little.
Wow.
So the bees was just one of those things that we're trying to also see if that works better than that or not too. I don't know about regards to other technologies. It's just one of those things where seagrass is a very fickle thing where if it gets If it gets stressed in any way, shape, or form, it usually doesn't do well. So that's why it's fine. It's difficult to strike that fine balance of what works and what doesn't.
Great, thank you. I got another question. They keep coming. Any findings yet from growing in the mixed meadow?
So I mean, right now we haven't done like any findings like after introducing the other two species, but what I'm hoping to see in the next like several months, even though we're kind of getting to the end of our growing season. So the growing season, for those who don't know, is technically the wet season as well. So from the early months of summer to about October, November, that's usually when we see the most growth in seagrass. That's usually it. The summertime is usually a very high energy time of the year for a lot of different things. So, for example, oyster spawning occurs during this time too. Mangroves spawn their copycools, et cetera, et cetera. So hopefully in the next summer, we'll actually see more of that like mixing of the species once we get into the growing season again. I'm not expecting to see any of that now that we're getting towards the end, but the amazing thing about seagrass is even during the non-growing season, the seagrass will still grow, just at a much slower rate.
Awesome, thank you. Unless there's any specific questions anyone wants to ask live, those are all the questions. We'll follow up with our guest who has the specific questions about the Loxahatchee manatee grass.
Mhm.
I want to thank Nicholas Curto. Thanks so much for sharing what's going on at Florida Oceanographic and our Seagrass Restoration Network.
Of
Thanks so
course,
much.
and please, like for any of the guests that are on here, for you, Khalida, or anybody, like I live here basically. I'm not gonna lie. I am always happy to show guests around to talk about all these different spaces as well, because I think it's one thing for me to talk about it, but it's another thing to actually see it in person and actually like walk through the front of house and see what's growing in these tanks. It's exciting to me. I know it's not the most exciting thing to everybody, but
Well, I can say I've spent time with you out there and it was exciting. We had a great time. And we'll be out there again. I'm coming up there tomorrow. Thank you, Nick, for sharing your time with us. And before we wrap, I've got a question about getting a replay. We will get that out. I want to try something before we all go. It's crazy time, and I want to take some lagoon yoga and bring it here on Zoom. I've used it for a couple of other things, and people have enjoyed it. Don't worry, no one's looking at you. You can just lower your eyes, close your eyes, take your hands, clasp them behind your head, elbows wide. Take a breath in through your nose, open up wider, lift your heart. Exhale, bring your elbows together, lower your chin to your chest. We're going to do this 2 more times. Inhale, open up, open oyster. Exhale, bring it all together, chin down, closed oyster. Last time here, open up, open oyster, heart forward, chin up. Exhale, bring everything in, elbows together, chin to your chest. I hope that moment of breath centered you a little bit and has you ready for the rest of your day. If anybody's interested in doing some lagoon yoga, a practice that we adopted from oyster yoga created at University of Central Florida, let me know. I've been doing it all summer and I'm happy to share. Since we're talking about seagrass, I love to add some oyster pose anytime I can. Thank you very much, everyone. Have a great day.
ah,